homenagem ao professor Ivan Izquierdo, neurocientista argentino, naturalizado brasileiro, que recebeu em 2007 o título de Doutor Honoris Causa pela Universidade Federal do Paraná e que infelizmente nos deixou em 9 de fevereiro de 2020, a Orquestra Filarmônica da UFPR irá apresentar o tango Poru na Cabeça, de Carlos Gardel. O arranjo é de Irina Lazareva.
Bom, um bom dia a todos que nos escutam aqui pelo YouTube, que assistem a essa reunião anual do Instituto Nacional de Neurociência Translacional. Antes de mais nada, eu gostaria de começar me apresentando. Meu nome é Sergio Ferreira e eu sou professor do Instituto de Biofísica e do Instituto de Bioquímica Médica da UFRJ, da Universidade Federal do Rio de Janeiro, onde eu coordeno, juntamente com meu colega Roberto Lent, que é professor do Instituto de Ciências Biomédicas da UFRJ, o INNT, o Instituto Nacional de Neurociência Translacional. Então, eu gostaria, em meu nome, do nome do Roberto, que infelizmente teve um conflito de agenda agora pela manhã e não pôde estar conosco aqui, mas estará conosco mais tarde um pouquinho, e de todos os meus colegas e nossos colegas, pesquisadores, técnicos, pós-doutores, estudantes de doutorado, de mestrado, de iniciação científica, estudantes de ensino médio, enfim, toda essa enorme equipe que constitui o INNT, o Instituto Nacional de Neurociência Translacional. Eu gostaria de dar as boas-vindas a todos e agradecer pelo interesse e pela presença. Eu acho que, falando do interesse que esse tema desperta, nós, através dessa divulgação que foi feita da nossa reunião anual, como nós já começamos a fazer assim em formato remoto desde a pandemia, nós tivemos agora nessa reunião, agora em 2022, que é a 8ª reunião anual do INNT, mais de 1.100 inscritos. Então, eu acho que isso dá uma medida muito boa do interesse, da curiosidade que o tema da neurociência desperta na população, sem razões para isso, porque a neurociência, afinal de contas, é o que controla, é o que permite que a gente entenda o que controla o funcionamento do nosso cérebro, o nosso sistema nervoso, o que controla o nosso organismo. Então, eu acho que é um tema que desperta muita curiosidade, muito interesse, e agradeço, então, pela presença de todos. Eu gostaria, então, de fazer aqui uma breve apresentação para os que não são ligados diretamente ao INNT, sobre o que é o nosso instituto, né? O INNT, ele foi criado há pouco mais de 10 anos atrás, dentro do programa dos Institutos Nacionais de Ciência e Tecnologia, lançado pelo Ministério da Ciência e Tecnologia. E esse programa é um programa bastante ambicioso que decidiu criar uma rede de institutos nacionais lidando com os mais diversos temas na fronteira da ciência, e alguns dos institutos nacionais se voltaram, então, para o tema da neurociência. O nosso, então, é um Instituto Nacional de Neurociência Translacional, e justamente a ideia dessa ponte aqui, que está nesse primeiro slide, que eu gostaria de mostrar a vocês, para os que não conhecem aqui a cidade do Rio, essa é uma ponte chamada de Ponte Rio-Niterói, que conecta as cidades do Rio de Janeiro e Niterói, a nossa vizinha aqui do outro lado da Baía de Guanabara. E, então, é uma ponte que conecta também a Universidade Federal do Rio de Janeiro, a UFRJ, com a Universidade Federal Fluminense, que também reúne vários integrantes do INNT. Mas, ao mesmo tempo, essa é uma ponte que tem um significado simbólico aqui, porque a ideia desse Instituto Nacional de Neurociência Translacional justamente foi criar uma ponte entre a pesquisa avançada em neurociência e a sociedade. Então, ele foi criado há um pouco mais de 10 anos atrás por um grupo inicial de 7 pesquisadores, reunindo cada um deles um grupo de pesquisas bastante amplo. Um deles era o professor Ivan Esquerdo, da PUC do Rio Grande do Sul. Nós acabamos de assistir a essa belíssima homenagem ao professor Esquerdo, que infelizmente nos deixou agora recentemente. Então, durante uma primeira rodada de 5 anos, a composição do INNT era desses 7 grandes grupos de pesquisa, com temas variados. E depois, na segunda rodada, a atual rodada dos INCTs, o INNT é composto, então, por mais de 20 pesquisadores, atualmente 20, começamos com 23, mas atualmente 20 pesquisadores que cobrem diferentes áreas do conhecimento. Então, eu gostaria de falar um pouquinho sobre o que é o INNT, para colocar essa reunião em contexto. Então, o INNT, a ideia justamente é construir essa ponte entre a ciência, a neurociência especificamente, e a sociedade. A ideia do INNT é, deixa eu colocar aqui um laser aqui rapidamente, é conectar as pesquisas, sejam elas pesquisas em laboratório, in vitro, com animais, eventualmente, com seres humanos, com a sociedade, então com avanços que possam se traduzir em melhorias no diagnóstico ou no tratamento das doenças, especialmente as doenças que atacam o sistema nervoso central, que afetam o sistema nervoso, o ensino e, eventualmente, também para o setor produtivo. Então, a ideia é conectar a ciência de vanguarda que é feita nas universidades, nos institutos de pesquisa em nosso país, com os usuários dessa ciência, que somos todos nós, a sociedade. Então, o INNT, na verdade, a ideia dele é conectar esses vários atores aí, né? Por um lado, os cientistas, pensadores e divulgadores que cuidam, dentro do INNT, da geração e da difusão do conhecimento científico. Esse conhecimento, então, pode ser utilizado como base por gestores, por políticos, por cidadãos da sociedade organizada, na proposição e avaliação de políticas, sejam elas de saúde, políticas econômicas, políticas educacionais, e, finalmente, a sua aplicação na prática, por professores, por médicos, por pacientes, por pais, enfim, por todos os cuidadores e atores no sistema de saúde, no sistema educacional e na economia brasileira. Então, essa é a ideia do INNT, construir essa ponte entre a neurociência e a sociedade. Por isso, o termo translacional, fazer essa translação do conhecimento dos laboratórios para a sociedade de forma mais ampla. O INNT está, então, distribuído em vários estados do nosso país. Aqui nós temos representados aqui, os 20 pesquisadores localizados, na verdade, no mapa brasileiro, os 20 pesquisadores atuais, que são todos pesquisadores classificados no nível de classificação de pesquisa do CNPq, muitos pesquisadores no nível 1A, vários também nos níveis 1B, C ou D, e alguns no nível 2. Esses pesquisadores estão distribuídos aqui entre 6 estados, com uma predominância no seu desenho atual no estado do Rio de Janeiro, mas com pesquisadores da Bahia, de Minas Gerais, de São Paulo, do Paraná e do Rio Grande do Sul. Esses 20 grandes grupos de pesquisadores se reúnem em torno de 20 linhas de pesquisa e em 4 grandes temas. E uma característica que eu acho que é muito interessante do INNT, e nós falaremos um pouquinho dela a seguir, ao final da minha apresentação, é o fato de que o INNT possui e criou o seu próprio programa de pós-graduação, que é o chamado Programa de Pós-Graduação em Neurociência Translacional, o PGNet. Bom, as 4 grandes áreas temáticas de pesquisa do INNT, então, são o desenvolvimento e a plasticidade do sistema nervoso, quer dizer, como começa a vida e como se desenvolve o sistema nervoso ao longo do processo de desenvolvimento embrionário ou pós-natal. A partir daí, estudos de neuropatologia, quer dizer, de doenças do sistema nervoso, tanto ao nível celular, das células que compõem o sistema nervoso, os neurônios, a glia, e quanto ao nível molecular, das moléculas que estão envolvidas nessas doenças do sistema nervoso. Isso permite, então, a formulação e o entendimento das bases para a compreensão das doenças neuropsiquiátricas, que são doenças muito prevalentes na sociedade, como, por exemplo, as demências, os transtornos de humor, a depressão, a apatia, a doença de Alzheimer, por exemplo, a doença de Parkinson, outros tipos de demências e o envelhecimento também, né? E, então, com base na compreensão então, dos mecanismos que levam a essas doenças neuropsiquiátricas, a proposição, então, de novas estratégias, novas abordagens clínicas ou diagnósticas para o tratamento, para o diagnóstico das doenças neuropsiquiátricas. Então, é uma abordagem bastante ampla, que vai desde o desenvolvimento até as doenças do sistema nervoso, como vocês podem ver. Então, rapidamente falando aqui de cada um desses 4 grandes temas, dentro da área de desenvolvimento e plasticidade do sistema nervoso, podemos ilustrar aqui, por exemplo, não são todas as linhas, mas, por exemplo, a linha que visa estudar a plasticidade dos neurônios e dos circuitos cerebrais, ou seja, como é que eles se modificam em diferentes circunstâncias fisiológicas ou patológicas, a sobrevivência neuronal na retina em desenvolvimento e malformações do desenvolvimento embrionário do sistema nervoso, quer dizer, como que alterações no desenvolvimento normal podem levar, no estado adulto, a doenças. No caso da linha de neuropatologia celular e molecular, o estudo de neuromediadores na retina doente, em doenças que afetam a retina, o estudo do papel de células gliais, células da glia, os astrócitos, a microglia, na inflamação e no déficit sináptico, nos defeitos sinápticos, na perda de função das sinapses, que são os pontos de contato e comunicação entre os neurônios, e estudos com células-tronco e inflamação em tumores cerebrais. Essa é uma área também bastante ativa, estudo de vários tipos de tumores cerebrais, como os astrocitomas, os gliomas, por exemplo. Depois, partindo então para a grande área temática que visa entender as bases das doenças neuropsiquiátricas, por exemplo, nosso grupo estuda mecanismos envolvidos nas epilepsias, na doença de Parkinson, na doença de Alzheimer, e estudando, por exemplo, dentro dessa última, como que alterações no metabolismo cerebral e a inflamação do cérebro levam à perda de memória, a perdas cognitivas na doença de Alzheimer. E, finalmente, na proposição de novas abordagens terapêuticas ou diagnósticas para doenças neuropsiquiátricas, podemos exemplificar aqui as linhas de pesquisa que visam entender ou desenvolver novas estratégias terapêuticas para epilepsias, como a reprogramação celular pode vir a ser empregada para o tratamento de doenças como a esquizofrenia e a identificação de novos biomarcadores para diversas doenças neuropsiquiátricas, por exemplo, a doença de Alzheimer. Então, só para dar aqui alguns exemplos, o INNT ele tem uma produção científica muito substancial. A cada ano são publicados aí cerca de 200 artigos científicos em periódicos internacionais, todos indexados e de bom prestígio em suas áreas respectivas. E eu citarei aqui apenas alguns poucos exemplos nas diferentes áreas temáticas. Então, na área de desenvolvimento, por exemplo, estudos no desenvolvimento fisiológico em xênopos, em embriões de xênopos, ou em mamíferos, por exemplo, aqui no cérebro de roedores, como que o cérebro se desenvolve e como que alterações nesse desenvolvimento eventualmente podem levar a transtornos no funcionamento cerebral, seja na infância, seja na vida adulta. Outra área que eu mencionei anteriormente, muito ativa no INNT, é a área de estudo de tumores cerebrais, tumores do sistema nervoso, e temos aqui, por exemplo, 2 exemplos recentes de publicações nessa área, entendendo o papel de transportadores específicos, moléculas específicas, mediadores de sinalização celular, como o ácido lisofosfatídico, por exemplo, na proliferação, na migração de glioblastomas, enfim, tumores muito agressivos do cérebro. Temos ainda estudos, por exemplo, do papel fisiológico de outro tipo celular da glia, que são os astrócitos, que têm um papel fundamental no funcionamento das sinapses, mas também estudos recentes vêm mostrando que não apenas para o funcionamento das sinapses, mas para a própria gênese das sinapses, para a formação dessas sinapses, em modelos in vitro e em modelos in vivo. Então, é uma área de bastante interesse de estudo também, compreender o papel desses outros tipos celulares, das células da glia especificamente, no desenvolvimento do sistema nervoso e no desenvolvimento da comunicação entre os neurônios das sinapses. Finalmente, exemplos aí de estudos na doença de Alzheimer, por exemplo, estudos envolvendo um mediador, um hormônio relacionado à atividade física na doença de Alzheimer, que parece ser um hormônio neuroprotetor, e estudos da sinalização por insulina cerebral, dentro daquele contexto de entender como alterações no metabolismo cerebral podem estar envolvidas na gênese da doença de Alzheimer. Então, essa é uma outra área ativa de estudos no nosso instituto. E, finalmente, eu gostaria de encerrar essa breve apresentação mencionando aqui, como eu mencionei lá no primeiro slide, eu acho, a existência do Programa de Pós-Graduação em Neurociência Translacional, o PGNet, que é coordenado de forma muito aguerrida e muito brilhante pelo nosso colega Vivaldo Moura Neto, que é professor emérito da UFRJ e é pesquisador do Instituto Estadual do Cérebro, Paulo Niemeyer, aqui no estado do Rio de Janeiro. O professor Vivaldo é o coordenador desse programa desde a sua criação em 2017. Ele foi criado, então, como um braço de pós-graduação do INNT. E a ideia é que ele é voltado exclusivamente para o doutorado, embora não seja necessário que o candidato ou a candidata tenha feito um mestrado anteriormente. É possível entrar na modalidade de doutorado direto também. Ele é atualmente sediado no Instituto de Ciências Biomédicas da UFRJ, como sede física, mas a ideia é que ele funcione, de fato, em rede. Então, ele é uma rede que tem orientadores distribuídos por muitos estados, até por mais estados do que os que compreendem aqueles dos pesquisadores do grupo central do INNT, nós temos orientadores de outros programas ainda, de outros institutos. E a ideia é que os candidatos, ao entrarem no programa de pós-graduação, no PGNet, possam vir a ser orientados ou co-orientados por 2 ou 3 supervisores diferentes que possam aportar à tese desse aluno ou dessa aluna experiências, expertises distintas e complementares, de forma a criar uma experiência muito rica de pós-graduação. E ele é um programa que prima pela internacionalização, porque os grupos de pesquisa que atuam no INNT e os orientadores externos também são grupos muito conectados com grupos de pesquisa em muitos países no exterior, né? Como nós temos aí alguns exemplos, mas faltam outros países aí, temos colaborações por todos os continentes, desde a Oceania, América do Norte, Europa, África, América Latina, naturalmente, enfim, Ásia. Temos colaborações em todas as regiões do mundo, em muitos países, em muitas instituições líderes de pesquisa nesses países. E eu não tenho certeza se esse número está absolutamente atualizado, mas já houve a conclusão de 8 teses e publicação de 42 artigos científicos por esses alunos que concluíram suas teses no PGNet. Então, é um programa que está se consolidando, ele é recente, é um programa muito novo, mas que vem se consolidando ao longo desse período. Então, é isso. Eu gostaria, então, de agradecer mais uma vez a todos que nos prestigiam aqui assistindo a essa apresentação. Somos agora há pouco eu soube que já eram 180 pessoas aqui no YouTube assistindo, afora alguns que nos assistem por outros canais, e é possível, então, antever que vai ser uma reunião muito rica, muito interessante, espero que traga muito conhecimento, muitas ideias, muitas perspectivas de interação entre todos os que assistem aqui a essa programação. Eu, então, gostaria de convidá-los agora para a próxima sessão, que se inicia daqui a 12 minutos, às 10 horas, uma mesa redonda coordenada pelo professor Roberto Paes-de-Carvalho, da Universidade Federal Fluminense, e nós teremos, então, um breve intervalo e depois o professor Roberto dará início aos trabalhos nessa nova sessão. Um bom dia e uma reunião muito produtiva para todos. Muito obrigado.
Welcome to this roundtable on the signaling pathways for neuronal and glial function. It's a pleasure to be here, and I want to first of all to thank very much the organizers, Ricardo Reis, who will be together with me in a moment, and Flavia Lima. And also I want to thank and congratulate our leaders in the INNT, Roberto Lent and Sergio Ferreira, for the excellent leading on this project. So the title, I want to welcome also and give good morning to everyone, and also good afternoon to the people in Europe now. It's a pleasure to meet you. I hope everything is okay. And the topic, as I said, is signaling pathways for neuronal and glial function. This is a very important issue, and we are going to have 4 small talks of 20-25 minutes, and I want to say that I hope you enjoy very much this meeting, and I will, as our time schedule is very tight, I will give a hint when 20 minutes is up for each of the speakers. And I also want to say that the questions that can be made by chat and by just asking for the people here, the speakers, will be at the end, or in the end of this round table. So thank you very much again. I hope everything is set. And I will give the word to Ricardo Reis, who will present the first speaker, who is Arturo Ortega. Thank you, Ricardo.
Thank you, Roberto. I would like to welcome everybody. Hi, Arturo. Nice to see you. I would like to give some words about Arturo Ortega, someone that I met 10 years, 15 years ago. Arturo is well known for his work in the neuroglia field. He is a chemist. He did chemistry in the 80s, and then he went to do a PhD in Israel when he cloned that day the kainate receptor. And Arturo, he is very well known for his work in not only neuron and glia cells. He used to work with the model of Bergman glia and the Miller glia in the cerebellum and in the retina. And Arturo is well known also for organizing several complex meetings. Arturo was the organizer of the last ASN, American Society for Neurochemistry meeting. And I met him also in several courses, international courses by Ibro and other ones. So Arturo, it's very nice to see you. I hope the pandemic finishes and we can bring you back to Rio again. And it's very nice to see you again. And thank you, okay? Obrigado.
Thank you, Ricardo. My screen is already working?
Yes, we can listen to you. Right.
Okay. So thank you, Ricardo, for the nice presentation. Thank you, Roberto, for the invitation. And I would like to share with you a very broad perspective of for years of work, what we've been doing with the glial glutamate transporters. And although my title refers only to GLAST, I will show you a couple of new data from GL21. Let me see, how can I do this? And then I can go with the laser pointer. Okay, I just wanted to show you this cartoon that I took from this paper, that I like it very much, because they say that the CNS can be regarded as a glutamatergic machine, and for somebody that has devoted 30 years to work with glutamate, this is a very nice slide. Okay, so and why we say so? Because it is well known that it's the major excitatory neurotransmitter, and therefore it is involved in most of the CNS functions. Okay. Glutamate exerts its actions by activating specific glutamate receptors, which are divided, as you all know, into ionotropic and apotropic receptors. I will not go into such detail this day, but I just want to tell you that when one decides to study this glutamatergic transmission, one has to find a suitable model. Although Ricardo already told you that I work mostly in cerebellum and in retina, and this is so because most of, for example, in the slide that I'm showing to you, you can see that parallel fiber to kingesal synapses in the granular molecular layer of the cerebellum is the most abundant glutamatergic synapse in the whole CNS, and this is electron microscopy photograph in which you can see the parallel fiber terminal, the protein cell, and you see all of these is surrounded by special radial glial cells, which are glia, which completely wrap the whole synapse, the molecular layer. We were able to develop a culture system consisting of cells, and we took the advantage to use a chip development in which we could, under standard conditions, get these nice cultures. This slide from the work of my previous time ago, I just want to show you the same thing, that this complete permanently surrounding. What I like very much is that you can see that even the proximity within the synapse is very, very narrow. So because of a big attack, therefore, it glutamate turnover needs the participation of glial cells. This is an old cartoon of the famous book Glutamate and GABA in the CNS. And just to remind you that what we know,
Arturo, can you listen to me?
glutamate shuttle, meaning that once glutamate is released, it's going to activate obviously its receptors that are in the neurons. Also,
nós estamos tendo problema de internet. A internet dele que está com falhas. Eu vou tentar ver se ele consegue desligar. Arturo, can you listen to me?
More than 90%. Yes.
Arturo, the internet is not very, it's not working. Can you, I think that if you shut down the camera, maybe the internet can get better. If you turn off the camera, just leave the presentation, perhaps we can have a better signal.
How do I do that? I will stop cam.
Yes. Maybe okay, now we can get, and you return to the presentation. Just click here.
This is present. And this is this is the screen. Now I cannot return. I can yes, like this. I cannot put it in the whole screen, but should it work like this? I cannot put it whole presentation.
The voice is better now. At least the signal, the internet got better now. Arturo?
I don't know how to. If I go back to the stream yard, I cannot. I turn it out and then I don't have the whole screen. But
Well,
I hope
okay.
that now it's working better.
Yes.
Can I continue?
Yes, please. Thank you.
Okay. So thank you. So in this slide, I just want to remind you that high brain functions such as learning and memory depend on synaptic efficacy, which involves the gene expression regulation of receptors, transporters, and ion channels. So today we're going to excitatory amino acid transmitters. The first two, GLAST and GLT1, are the glial transporters, although GLT1 has also been shown to be expressed in some sort of specialized neurons, mainly in the hippocampus and a little bit in the cerebellum. But in any event, those are the major glial glutamate transporters. So using these chick cerebellar Berman glia primary cultures, some years ago with Marco Gonzalez, we described that pre-incubation with glutamate would decrease, and then washing the glutamate would decrease the uptake activity. And at that time, we found out that this was not a receptor-mediated event because it was not mimetized by kainate, AMPA, or NMDA. But something that we found out at that time and we couldn't explain, it was that the glutamate effect was completely abolished by staurosporin. So that was a piece of evidence that at that time we couldn't figure out. In any event, we continued the characterization and found out that we had a decrease in the activity that is shown here. Okay, that long-term TPA stimulation would almost kill the whole activity. That we had a decrease, a time-dependent decrease in the amount of the protein. At that time, we didn't do biotinylation, but we did a whole thing. And with Esther, we found out, with Esther Lopez, we found out that the mRNA would decrease in dose and time-dependent fashion. Later on, also with Esther and with Sandra Rodriguez, who unfortunately she just passed away last year, we found that we characterized the promoter region, and we found out that the whole glutamate responsive element was restricted to a region of I would say like 20 or more than that, 40 kilobases, in which we were able to detect a YY1 binding site, Yin-Yang1 binding site, and therefore when we expressed, we overexpressed YY1, we could see that we shut down the transcriptional activity and also the amount of mRNA. More recently, we started to figure out what would be the mechanism of action of this repression, and I had this enthusiastic graduate student that I have it in a picture. Her name is Ada Rodriguez Campuzano, and she came to me and she told me, you know that YY1 is known as member of a polycom complex that it's able to recruit DNA methylase 3, and therefore it's probably we have a dynamic methylation in the promoter. So what she first did was to evaluate the amount of 5-methylcytosine as an index of methylation, of DNA methylation. And when she went with D-aspartate, which we should remember, this is the way to activate or to bind to the transporter, because aspartate has very low affinity towards the receptor. So what she found, and with glutamate, that there was an increase in 5-methylcytosine, not too much with 100 micromolar D-aspartate, a little bit of response. But with VPA, which is valproic acid, which is an inhibitor of histones deacetylases, we have an increase in 5-methylcytosine. There, we decided to measure the uptake activity, and under hypomethylation conditions, when we compete for the DNMT3 with 5-ASA, 24 hours, we have a very nice increase in the uptake, meaning that if you prevent the action of DNA methylases, you have an increase in the uptake. And what I show here is that she reproduced some of our earlier experiments, meaning that YY1 would have to be in the nuclei upon TPA, which is an activator of protein kinase E under glutamate, and a little bit under D-aspartate. Okay, but this is a result that was really, I think that I discussed with Ada to perform it for several times, because what she found is another time, you prevent DNA methylation, dynamic DNA methylation, you have an increase in the uptake. But when she went with a blocker of GLT1 in these cells, in Berman glia cells, the expression of GLT1 is almost non-detectable. It's very low. Almost 90% of the uptake activity is not blocked by DHK. But under hypomethylation conditions, she found that, okay, of this increase in the uptake activity, 16% came from GLT1. And she went to take a look at the GLT1 levels, and she found out that under prevention of methylation, she had a very strong signal, meaning that in these cells, in Berman glia cells, you have an increase in GLAST expression, or you have mostly GLAST expression, because GLT1 is methylated. And this is and that you can this is an activity dependent phenomenon. Okay. But all what I've been talking right now, it's long-term modulation. That would be like 12 hour, 24 hour. But if we're speaking about the glutamate, glutamine shuttle and astrocyte lactate shuttle, meaning interaction in the minute level to try to understand how glia is involved in parallel fiber Purkinje cell synapses, there has to be another level of regulation. So with Marco Flores, what we found out was that upon stimulation of the transporter with 1 millimolar D-aspartate, we had a decrease in methionine incorporation, meaning that there was a decrease in protein synthesis. And just to make the long story short, translational control, it's another like fine-tuning of protein synthesis control, and that it has a fast response, and it depends. It can be general, and it can be specific. So with Ileana Barrera, what we found out is that not only we were able to show that the initiation of translation, the recruiting of the first tRNA and formylmethionine was prevented through phosphorylation of eukaryotic initiation factor 2A, and that this was time and dose dependent. But also, we found out with Sila Martinez that after the initial decrease in the initiation process, we also had a phosphorylation of the elongation factor 2, which prevents the translocation of the ribosome, meaning that this decrease in protein synthesis is transient, and that it had to do with the uptake process. Okay? And therefore, Sila asked herself how this is going. If the uptake of glutamate or even aspartate is a sodium-dependent process, then sodium might be the signal to trigger all this cascade of events. So she decided to measure calcium influx, and she found out that upon D-aspartate, she had a very nice calcium influx, which was prevented by a blocker of the sodium-calcium exchanger, meaning that the transporter, GLAST, would be a signal transducer entity, because through the entrance of sodium would activate the sodium-calcium exchanger, and this would lead to protein synthesis reduction and some other signal transduction pathways that we were able to characterize later on. And if we were talking about protein synthesis regulation, we wanted to see whether mTOR, which is the master regulator of protein synthesis, was involved in the phenomenon, and Edna Suarez demonstrated that this is indeed the case, that the activation of GLAST transporter leads to mTOR phosphorylation, and obviously to the phosphorylation of one of their downstream factors, like for elongation for EVP. This is the factor that hijacks the translation factor for initiation translation factor 4E, and therefore prevented protein synthesis. And this would make sense, and as you can see, this is a D-aspartate mediated effect that is due to the transport phenomena, because it's mimicked by this transportable blocker like THA, and that is prevented by mTOR complex 1 blocker like rapamycin. So we have so far we have demonstrated, or we were able to demonstrate, that the major uptake system in Bergman glia cells is GLAST, that that GLT1 is not expressed because its promoter is likely to be methylated, which I think that this was also found in hippocampus, that there was a regulation of GLT1 methylation, and that GLAST would function as a signal transducer element to control protein synthesis. Then we went to look if everything was correct. Then we went to look for the ribosomal profiles, and with Reinaldo Tiburcio, we found out that these are the ribosomal profiles. I just want to show you that the first peak is the monosomal fraction. Then you have the polysomal fraction. If you have a very strong protein synthesis, you would expect most of the mRNAs would be in the polysomal fraction. So what Reinaldo found that was exactly when we had a decrease in protein synthesis, we had an increase in RNAs in the monosomal fraction, meaning that translation was not as efficient as it should be. And then after 30 minutes, he found out that all these RNAs were returned to the polysomal fractions. When we were trying to find out what does these results mean, we found out that there are certain RNAs which have very complex 5' UTRs like glutamine synthetase. And with Daniel Martinez, remember glutamine synthetase is very important because part of the glutamate that is taken up by the glia cells is going to be converted to glutamine, and this is going to go back to the neurons via the SNADs. So what he found out that upon glutamate exposure, there was an increase in glutamine synthetase synthesis, and therefore, with Reynaldo, we went to check another time the ribosomal profiles, and we PCR'd the several fractions that we had in the polysomal fraction, and we found out that after 30 minutes of glutamate exposure, most of the in the polysomal fraction, we have the glutamine synthetased mRNA enriched, meaning that here we have that was the activity, and this is the polysomal fraction, and you see in 15 minutes we have we have a very nice enrichment of glutamine synthetased mRNA. Okay, so with that, I think that I will finish here, and I will just go with the model that we are working with, meaning that once we have this glutamate that has been released due to the remember, like in retina and in the cerebellum, we have a permanent tonic activity. So this glutamate is going to be taken up by GLAST, and this will be transformed to glutamine through glutamate synthetase. Since GLAST is going to increase sodium levels, these sodium levels are going to activate the sodium-calcium exchanger, and by this means would downregulate in the short term protein synthesis, both at the level of the initiation, but also at the level of elongation. And this allows that mRNAs with very complicated 5' structures like the glutamine synthetase mRNA would be able to bind initiation factors and could be actively translated. And we were able to show that upon glutamate exposure or deaspartate exposure, the mRNA for glutamine synthetase migrates to the polysomal fraction, and that this results in an increase in the synthesis of the protein and an increase of its activity, favoring the existence of the glutamate-glutamine shuttle, which I should say has been questioned by several authors. And with all this, I just want to acknowledge the people that made the work, and my dear colleague Esther Lopez-Baigen, and also Clara Hernandez, which is my right hand in the lab, and CONACYT for funding this all these years the lab, and also Fundación Pandea. Thank you.
Okay, thank you very much, Arturo, for this wonderful presentation. And I hope you the questions will be in the end. Okay.
Okay. Thank you.
Thank you. So our next speaker will be Dr. Hannes Schmidt from the University of Tübingen. Hello, Hannes. Nice to meet you. Are you hearing me?
Yeah, I can hear you. Can you hear me?
Okay, we'll present you now. So, yes. So, Dr. Hannes Schmidt is a lecturer at the Interfaculty Institute of Biochemistry at the University of Tübingen in Germany. Using molecular genetic and imaging approaches, Dr. Schmidt studies the development of axonal projections that shape the complex circuits of the nervous system. His research contributed to the identification of a cyclic GMP signaling cascade that controls axon bifurcation in sensory neurons. After his diploma thesis at the Max Planck Institute of Psychiatry in Munich, Martinsried, and Schmidt graduated in biology from the University of Leipzig in 1995. He earned a doctorate from the Free University of Berlin in 1999. After a postdoctoral period at the MRC Center for Developmental Neurobiology at King's College London, funded by a Marie Curie Fellowship of the European Commission, he moved to Fritz Hatchen Laboratory at the Max Delbruck Center for Molecular Medicine in Berlin-Busch as a project leader in the Collaborative Research Center Developmental Disorders in the Nervous System. In 2011, Dr. Schmidt habilitated at the Free University Berlin in zoology with the topic Signaling Mechanisms of Axonal Guidance and Branching. In 2016, Dr. Schmidt assumed his current position in the Department of Signal Transduction and Transgenic Models, headed by Robert Feil at the Interfaculty Institute of Biochemistry in Tübingen, the University of Tübingen. He is also a member of the German Society for Neuroscience, and his research supported by the German Research Foundation. So thank you very much to accept our invitation. Go ahead, and I will give a hint at 20 minutes of presentation, okay? Thank you.
Thank you, Roberto. Thank you very much for inviting me, and thank you to the organizers from the INNT. So bom dia from Tübingen, which is situated in the southwest of Germany. I hope you can see my presentation. Can you see it?
You can go ahead.
Thank you. So my talk will center on a cGMP signaling pathway that controls sensory axon bifurcation. cGMP is a classic second messenger, cyclic guanosine monophosphate, which is kind of modularly organized, so it can be activated by different pathways. But what I'm interested specifically in is the cGMP signaling pathway activated by C-type natriuretic peptide. Okay, I can't skip to the next slide. Yeah, there is a technical problem. I can't proceed to the next slide.
Hi, Ricardo, do you know what's happening?
Qual foi a pergunta, Roberto?
Perguntei por que ele não está conseguindo botar o slide para frente.
Cara, não sei.
Just a moment.
Can you go back in the, not the full presentation, with the whole sequence?
What do you mean by the full sequence?
I know. All your slides, because you have just the first slide here. If you remove the full presentation mode, if you have all one by one, I don't know why the second is not coming after the first.
Okay.
So
can you now see the second?
Yeah. Can you put in the sequence where we can see all the slides, just to see why the second's not coming after the first?
Yeah, here are the slides.
Because I see only the first one, CNP-induced cyclic GMP. This is PowerPoint, right?
This is PowerPoint, yes. So here are, so on my monitor, I can see all the slides.
Well, this is the point. I can see only your first one. I don't know why I can see only the first one and I cannot see the sequence of all your slides.
I don't know why this is. So I will go out of the presentation and start it anew.
Yeah, maybe this is the best way to do. If you start all over again.
Can you see it?
No, I just see you. If you can share now, let's see it now. Yes, now I can see the second slide. Brain functions. All...
Sorry for that.
Okay.
So
thank you. Yeah, now it's all right. It's perfect.
Thank you. So just for an introduction, I want to make you aware that all the sophisticated functions of our nervous system, which are, of course, rooted in the genes and then in the proteins, which organize different cells in the nervous system and lead then to specialized circuits that lead to behavior, are all rooted in the hallmark morphology of the neurons, which is very asymmetrically organized and has these highly branched dendrites, the soma, and one axon, which can also be branched. And the function of all our daily life, the interaction with our environment, but also the detection of inner functions relies on the fast and precise propagation of electrical impulses within these neural circuits. However, not so much is known which molecules actually initiate the branching and the formation of this circuitry. And the nervous system development has different stages. So we heard about the process of synaptogenesis in the talk before by Dr. Ortega, but I'm interested in this stage here, neurogenesis, where neural outgrowth occurs. And we want to investigate which mechanisms contribute to the establishment of neural connections. And when the neurons are formed, there's first a process of neuronal polarization. This means the single axon is determined, which then grows. And this growth is guided by certain guidance molecules, which can be attractive or repulsive in their nature and can be mediated by a contact or by a diffusible gradient. I think the four best well-known families which contain these guidance factors are the netrins, the semaphorenes, the slit, and the efferents. But there's also the process of axon branching and presynaptic differentiation, which means that you have a structural basis for the distribution of information to different parts of the nervous system. And this branching can be regulated either in an activity-dependent process or independent. And basically, there are two modes of axonal branching, gross cone splitting and interstitial branching. And all these processes rely on the outgrowing axon, and at the tip you have a very motile structure, the so-called growth cone, which acts as a navigation system of the neural outgrowth. And there, cytoskeletal proteins like microtubuli or actin are distributed in this growth cone, and their rearrangements finally decide about the direction of growth, but also in the case of axon bifurcation, in the process of axonal branching. And the questions which are investigated in this circumstances are which molecules actually and signaling pathways downstream of the molecules initiate neural arborization. Is there kind of a common signaling mechanism for all forms of branching? How is branching coordinatively regulated with neural growth and guidance? And how is neuronal activity involved in the establishment of this branching pattern? And our lab in the last years investigated the question which molecules and signaling pathways are involved. So as I said, there are basically two modes of axonal branching. One is related to the activity of the growth cone. It's mostly a bifurcation where the growth cone splits up and then forms two daughter axons with growth cones. And the second process is a process of interstitial branching, where collaterals sprout out from the axon shaft and then establish new pathways. However, more knowledge is needed about the molecular mechanisms which are involved in the generation of these forms of branching. And the investigational system which we used is the embryonic spinal cord, where in the DRG at both sides of the spinal cord, the cell bodies of the sensory neurons are located, which transmit pain, information, but also information about the tension of muscles or temperature. And they have all a very stereotyped branching pattern. So the central axons of these neurons grow towards the spinal cord through the so-called dorsal root. And when they arrive there at embryonic days 10 to 13 of embryonic development, they split up and bifurcate, which is shown here in A. And then two daughter axons extend at the dorsolateral margin of the spinal cord in opposite directions. And after a waiting period from one day, then the collaterals are actually formed from these daughter axons, which then penetrate into the embryonic spinal cord and terminate there in different layers of the cord, depending on their sensory modality. So the nociceptive neurons, for instance, terminate here in the dorsal horn, whereas the mechanoreceptive neurons more terminate in ventral regions. And there, in their destination areas, further terminal branching can occur. And we use this system to investigate whether cGMP signaling components have a function in this. First, a short introduction to cGMP signaling. You see here its structure. It's a cyclic nucleotide like cAMP, which is pretty much everywhere in the organism. And cGMP is generated by two families of generator proteins. This is on one hand the so-called soluble guanylyl cyclases, which are activated by nitric oxide, a gas which can easily penetrate the cell membrane. And on the other side, the family of particulate guanylyl cyclases, which are activated by ligands like the natriuretic peptides or guanylene and uroguanylene and heat-stable enterotoxin. So you have two different kinds of generators of cGMP, and then downstream of the cGMP, three different types of effectors, which can be either phosphodiesterases or protein kinases, which are cGMP dependent, and then a family of cation channels, which are also activated by cyclic nucleotides. And when we started the project, we didn't have a situation like now that we can get all these expression information from databases on the net where single-cell sequencing data are available. We did it ourselves by an RT-PCR screen and just checked which of the generators of cGMP are actually found in the embryonic DRG neurons. And we did this with cDNA from spinal cord and from DRG, and for the soluble guanylyl cyclases, but also for the NO-generating enzymes, NO synthase 1 to 3, there was not much expression in the DRG. For the case of the particulate guanylyl cyclases, we had two hits from these particulate guanylyl cyclases. One is NPR2, the natriuretic peptide receptor, which is very often also called guanylyl cyclase B, and guanylyl cyclase E. On the activators of cGMP signaling, we found that CNP is expressed in the DRG and in the spinal cord. And this was interesting because CNP is a ligand for NPR2. And we elongated this screen also for the effectors and found that cGMP-dependent kinase 1 is also expressed there. So these three could make a good signaling cascade with a ligand, the receptor, and the kinase, which could be involved in the function. To seek further evidence for CNP-induced cGMP signaling, we seeked for more biochemical evidence. But first, I want to introduce you to the two molecules. C-type natriuretic peptide is a 126 amino acid long peptide which is further cleaved, for instance, by the protease furin in its active forms, CNP53 and CNP22. At the C-terminus, it has a classical structure of a disulfide bridge, which forms then a ring structure. And it's known that it's mainly a paracrine or autocrine effective. And CNP has two receptors. One is NPR2, which can generate cGMP, but the second one exists also, which is NPR3, which is called a scavenger receptor because it just mops up superfluous CNP, but has not cytoplasmic guanylyl cyclase activity. The natriuretic peptide receptor 2 itself, which is also called GCB, is a transmembrane protein of 1047 amino acid. It's mostly acting as a homodimer, and the binding of CNP to NPR2 activates cGMP synthesis by its guanylyl cyclase domain. It has also a so-called kinase homology domain, which is regulated by the phosphorylation of several serine and threonine residues. And a dephosphorylation of these residues leads to a desensitization of the receptor guanylyl cyclase. It's found in brain, lung, bone, heart, and ovary tissue. And this CNP NPR2 signaling is most prominent for its function in bone growth, but also in oocyte maturation and aortic valve function. So its function in bone growth is also documented in the phenotype of the knockout mice for CNP and for NPR2. So they are shorter than their wild-type litter mates, have also an increased postnatal mortality. And the impaired scavenging of CNP in mutants of NPR3 can lead to an overgrowth of bones, and which is documented by this hump in the mice. And in human, there's also known that mutations in the NPR2 gene on chromosome 9 can lead to an autosomal recessive skeletal dysplasia, but it's very rare. It's only found in 1 in a million live births. And these patients suffer from disproportionate short limbs and spine, as depicted here. And also heterozygous carriers of these NPR2 mutations are shorter than the average population. Okay, but coming back to the nervous system, so we seek this biochemical evidence for CNP-induced cGMP signaling in the DRGs. And one way to test this is to use the established phosphorylation substrate of the cGMP-dependent kinase 1, VASP, a member of the ENA-VASP family of actin regulators, as a monitor to test this, because phosphorylation of serine 157 in VASP induces a mobility shift, which can be detected in Western blot. And when we applied CNP, indeed, we saw an increase in phosphorylation as compared to the negative control. However, stimulation with NO donors did not lead to an increase in the phosphorylation of VASP. Also, the natriuretic peptide type A, which binds to NPR1, did not induce phosphorylation of VASP in comparison to CNP. And we could show that this cGMP signaling pathway indeed proceeds via NPR2, its receptor. We used a loss of function mutant of NPR2, which is called NPR2CN, which carries a spontaneous mutation in the guanylyl cyclase domain, which leads then to a loss of function. And we could show when we stimulate with a stimulator of CGK1, 8-PCPT-cGMP, we could see an increase in phosphorylation of VASP in the wild type, and also in the NPR2 loss of function mutant. But when we stimulate with CNP, which is a ligand for NPR2, then this leads only to phosphorylation in the wild type, but not in the NPR2CN loss of function mutant, as shown in the last lane. And we could confirm that this proceeds via CGK1 because the application of CNP in CGK1 mice did also not lead to an increase in phosphorylation. So we had evidence that CNP signaling indeed is functional in DRGs. We also used real-time thread recording of cGMP in DRG neurons, for which we have the cGPNC sensor CGI500 transgenically expressed in mice. And the mechanism is shown here. So you have two fluorochromes which are coupled by a cGMP binding domain. And if these fluorochromes are in close distance, then the activation of the left fluorochrome leads also to emission of fluorescence from the right fluorochrome. However, when cGMP binds to this sensor, then the distance increases and there is no transfer of thread energy anymore, as shown here in this example. So you see the fluorescence of both fluorochromes. And when cGMP binds, then the CFP fluorescence goes up and YFP fluorescence goes down. And if you look then on the relation of the two fluorescences, you can get a measure for cGMP concentration. And we did this with DRG neurons, which were isolated from the CGI500 sensor mice, and could show that only CNP, but not DANO or ANP or BNP, leads to an increase of cGMP in these DRG neurons. We generated LAC-Z reporter mice for both the ligand CNP and for the receptor to map their expression because good antibodies to both proteins are not available. And we could show that in the time window when bifurcation actually occurs, expression of both proteins starts in the mouse embryo. So the expression of the ligand CNP precedes a bit the expression of the receptor, and you see that the ligand is expressed in the spinal cord, whereas the receptor is found in the DRG, but also in the craniosensory ganglia, and the ligand is also found in the hindbrain. Please keep in mind that these LAC-Z constructs carry a nuclear localization sequence, and therefore you see the expression only in the nuclei of positive cells. And the same could be used for staining transversal sections, and you see that the ligand CNP is found in the dorsal quarter of the spinal cord, of the embryonic spinal cord, whereas the receptor is found in the neurons in the dorsal root ganglia. We could also use an antibody against beta-gal to show this expression, and this confirms the LAC-Z staining, but this can be coupled with a double staining with an antibody against the third component of the signaling pathway, the CGMP-dependent kinase 1 alpha, and you see that you have an overlap of CGK1 alpha with the NPR2 expressing DRG neurons, because now all the cells are stained in their complete extent. You see also the axons which form the dorsal funiculus at the side of the spinal cord, where they come in close contact with the CNP secreted here in the dorsal spinal cord, which is indicative for an instructive function of this signaling pathway. We also showed that the same complementary distribution of the ligand and the receptor together with the kinase is found in the craniosensory ganglia and in the hindbrain, but to this I come later. To analyze now whether there are differences in the branching of these DRG neurons, we used three different methods. One is the labeling of DRG neurons with a lipophilic dye I, which can be injected with a glass pipette and then just stains the whole neuron. And then you can look from the dorsal side to see the pattern. And if you have a higher magnification, then you can see in the wild type that these DRG neurons really form a nice T or Y-shaped bifurcation. Other techniques would be the use of a Cy1 reporter mouse line generated in the lab of Joshua Sainz and Jeff Lichtman, where only a minority of DRG neurons expresses the YFP reporter. And this can be used then to analyze the trajectories of single neurons, as shown here. So in the wild type, you have this T or Y-shaped bifurcation. And in a mutant of NPR2, which is called 7A, because the 7 serine threonine residues in the kinase homology domain are mutated to alanine, you see that bifurcation is completely lost, and these neurons only turn in one direction. The third way of showing the bifurcation behavior of single DRG neurons is a conditional alkaline phosphatase receptor, which has a very low recombination efficiency in combination with an appropriate creline. We used here a creline which is inducible by tamoxifen under the control of the NPR2 promoter. And due to the low efficiency, you can see then really only one single axon out of maybe 5,000 neurons in one DLG. And if you compare here the heterozygous situation, then you see the bifurcating neuron, and in the case where both alleles are mutated of the NPR2 gene, then you have only the turn, and we quantified this over all spinal levels from cervical to sacral regions and found that in the wild type we have mainly 100% or nearly 100% of bifurcating neurons, but in the mutated alleles, we have only turns which either are in the anterior or posterior direction. And the same was found also here is shown for di labeling for CGK1, but also for the ligand CNP, that axon bifurcation is completely lost if you interfere with this CNP NPR2 CGK1 signaling pathway. As shown before, we registered also that there is a complementary distribution of the ligand and the receptor and the kinase in the hindbrain and in the cranial sensory ganglia. Okay, I will come to an end. And we could show then with this NPR2 CREAT2 mediated recombination system that also these neurons here, as an example, the trigeminal neurons, lack the bifurcation which is found in the wild type in the heterozygotes when both alleles of NPR2 are mutated. And this leads then in the mutant also to a decreased diameter of the trigeminal funiculus in comparison to the wild type. Interestingly, the second type of branching, the collateral formation, is not impaired, so there is no difference in the density of collaterals between the NPR2 heterozygous and homozygous mutants. And also the peripheral projections are not different between the genotypes. Summing up, we identified a signaling pathway with the components C-type natriuretic peptide, the receptor guanylyl cyclase B or NPR2, and the cGMP-dependent kinase 1-alpha, which is important for the bifurcational branching of DLG neurons, but not for the collateral formation, as shown here. And this signaling pathway regulates the bifurcational branching of both DLG neurons and cranial sensory ganglion neurons. So we'll now check whether also other neuronal populations will use this signaling pathway for their branching behavior. The second type of branching, which DLG neurons express the interstitial branching, is not affected by changes in this signaling pathway. And what I didn't have time today to show you is that on a functional level, this loss of sensory axon bifurcation results then in decreased pain perception in conditional mutants of NPR2. At the end, I want to show the collaborators. So this work was mainly done in Berlin-Buch at the Max Delbrück Center for Molecular Medicine, where different groups collaborated. The group of Fritz Ratjen, Gary Lewin, the group of Carmen Birchmeier and Michael Bader to do this. But for the pain studies, we collaborated with Aaron Schmidt from the University Witten-Herdecke, now in Frankfurt am Main. The CGK1 knockout mice were donated by Franz Hoffmann from the Technical University, and the cGMP FRET measurements are done at the University of Tübingen in the lab of Robert Feil. Thank you very much for your attention.
Thank you very much, Hans, for this very interesting presentation. And as you know, the questions are appearing here in the chat. And it will be in the end. Okay, thank you very much.
Thank you.
We are going to move to the next speaker. Which is, I don't know. I think you can, yeah. The next speaker will be Dr. Renato Socodato. Hello, Renato. How are you? Thank you to accept this invitation, and I will present you. I hope everything is okay with your presentation. Is okay? The slides are okay? Well, I will present you. So Renato Socodato is Brazilian, first of all. Obtained his PhD in neuroscience in 2012, working in my lab. So my pleasure is a double pleasure at the program of neuroscience of the Fluminense, and working in the signaling of cytoplasmic tyrosine kinases and regulating death and survival mechanisms during neuronal development. During his PhD, he became interested in tyrosine kinase signaling, controlling microglial cell function in the CNS, focused on the retinal tissue. In 2014, he joined the Relvas lab as a postdoc fellow at IBMC in the University of Porto, in Portugal. He integrated the research team to study the molecular mechanisms leading to microgliosis and associated neurodegeneration in the CNS. In 2019, he became a research fellow funded by FCT and allocated in the Relvas lab to study the interplay between microglia, neuroinflammation, and neurodegeneration. His recent publications show critical roles for tyrosine kinase and RhoGTPase signaling in controlling microglia-dependent inflammation and neurodegeneration. He is particularly interested in classical RhoGTPase signaling, HOA, HAC1, and CDC42, and the crosstalk between microglia and synaptic dysfunction, including steady state and neurodegenerative conditions. He is now a postdoctoral fellow at the Institute for Molecular and Cell Biology of the University of Porto. So thank you very much again. Renato, you can go ahead. I'm going to give you a hint in 20 when you complete 20 minutes, okay? Did you hear me? So you can present. Use the full screen. They are saying here that use the full screen or slide. Renato, can you hear me? No. Yes, I'm not hearing you. Just a moment, please.
Can you hear me now?
Yes. Okay. Yes. Okay.
Let me know if the presentation.
You have to put your presentation now. Okay. Okay.
It's showing.
No, not yet. No, not yet.
I'm going to share screen. That's it.
Yes. Yes. I think so. I think so. Share screen, yes. Share screen, yes.
It's working.
No. Share screen, full screen. Not yet. Not yet. It's giving a neck. It's giving a neck.
Like share screen? No, share screen.
It's okay now? No.
No. Can you see the shot? At the bottom of the screen, you will see the option share or present.
Uh-huh.
Click that.
Yes, but I don't know if it works. It says that my browser cannot compute. I will just leave and enter again and see if it works.
Yes.
Vou tentar de novo.
Pode ter sido o mesmo problema que aconteceu com o Ramos. Talvez você tenha que voltar e entrar de novo. Não.
Acho que sim.
PowerPoint está aberto?
Assim.
Clica em Share.
Espera aí.
Vai funcionar.
Vai, tem que ser.
Doutor Renato,
Sim.
o senhor consegue abrir o seu PowerPoint? Fora daqui do YouTube. O senhor vai abrir o seu documento, seu arquivo de PowerPoint.
Sim, tá abrindo.
O senhor vai aparecer lá embaixo para o senhor apresentar.
Só um minuto que ele tá abrindo.
Abriu?
Sim, tem lá um modo de apresentação.
Isso, perfeito.
No PowerPoint?
No PowerPoint.
Isso. E daí o senhor volta para cá, para a tela, e o senhor vai colocar no modo apresentar.
Eu ponho no modo apresentar no PowerPoint?
Não. Aqui, o senhor vai voltar aqui para a nossa tela.
Sim.
Conseguiu voltar aqui para a tela principal?
Sim, põe Present.
Isso. Compartilhar tela.
Sim.
E daí o senhor pode colocar a tela cheia. É só clicar em cima da sua tela e compartilhar. Não tem problema. Ou o senhor pode compartilhar uma janela, que seria o seu PowerPoint. O senhor consegue
porque quando eu
encontrar a tela cheia?
ponho compartilhar a tela cheia, ele me manda uma mensagem a dizer que o Google Chrome perdeu a permissão de captura de tela.
Entendi. O senhor consegue, então, compartilhar a sua janela?
Vou voltar e vou fazer. Compartilhar a janela. Window.
Isso.
PowerPoint.
Seleciona o seu PowerPoint.
Assim, continua dando o mesmo problema.
O senhor pode fazer uma sugestão, passar a apresentação para o próximo, e daí o senhor envia a sua apresentação para mim por e-mail, que eu consigo apresentar aqui para o senhor. Pode ser?
Pode, só que minha apresentação não consigo mandar por e-mail. Tem que ser tipo por um e-transfer, alguma coisa assim.
Pode ser pelo e-transfer.
Tá bom. Me diz o teu e-mail que eu mando já.
Já enviei aqui. Obrigado, doutor Renato.
Então, o que nós vamos fazer? Passar para o próximo? Vou esperar um pouco. O próximo sou eu mesmo. Então, vou passar a palavra para o Ricardo nesse momento. Obrigado.
Obrigado você, Roberto. Só avisar o pessoal que são 4 palestrantes em 4 lugares diferentes no mundo. Isso acontece, pessoal, mas a gente vai conseguir terminar as palestras com a qualidade que está sendo apresentada. Então, eu gostaria de apresentar o professor Roberto Paes-de-Carvalho, que é meu amigo. Ele tem trabalhado com neuroquímica desde que eu entrei novinho para fazer mestrado. Roberto é médico. Ele fez mestrado e doutorado no Fundão, na UFRJ, e desde a década de 80, Roberto já criou um grupo na Federal Fluminense. Naquele primeiro slide que o professor Sergio Ferreira mostrou, aquela ponte leva ao laboratório dele lá na Federal Fluminense, e Roberto sempre se preocupou. Então, vou mudar para o inglês agora. Roberto is working since the 80s with the retina as a model to study neuron-glia interactions. So the subject of this table today is exactly to match how glia and neurons interact, and Roberto has done a supreme job doing this. I'm sure he's going to show excellent data here. And Roberto, thank you for the opportunity to present you.
Thank you very much. I'll wait for the presentation. Okay. I hope you are hearing me well. Please, Ricardo, tell me when I have 20 minutes. And just a moment. Okay, so the title of this presentation is AKT regulation by nitric oxide and calcium in developing retinal cells. So we are working in the Department of Neurobiology and Program of Neuroscience of the Fluminense Federal University. So the same thing is happening. First of all, I want to show the retina and to honor. It's an opportunity to honor Santiago Ramon y Cajal for his beautiful drawings. And here you can see the retina with the few cell types, and in one of his wonderful drawings. So the retina, and here the next, you can see the retina of the chick, showing the different layers, as you can see here. I don't know if you can see my mouse, but you can see here different layers of the chick retina, which is very similar to other structures of the central nervous system. And when you have layers of cell bodies separated by layers of plexiform plexus layers, and we want to say that the retina is very interesting, but we use as a model for CNS neurochemical studies. So the next, the cheek embryo retina is a structure of the CNS, of the central nervous system, very useful as a model for developmental and cell biology studies. So you can see here the embryos growing, and you can see them with the big eyes, and so you can get the retina. It's very easy to get it, and it's taking longer to go from one slide to the other. Okay, as you can see here, the very early embryo, so embryonic age 8, you have the embryo here with the big eyes, and can remove dissect the retina, and the retina has this appearance with no layers, and you have only neuroblasts here. And the difference from E18 is that you have a characteristic mature layers of the tissue you can see here. So also very important as a model that you have next. The neurogenesis of the retina is very well known. You can see the sequence of the appearance of the cells in the tissue, and this is very common to many species of vertebrate species, as you can see here, and also in the chick. And so the chick retina, one of the advantages of the model for us is that you can remove from the E8 embryo and make cultures, and different cultures can be made. So we have here the mixed cultures and containing neurons and also glial cells that have flat cells here and the glial cells, and you can also obtain purified cultures of neurons as well as glial cells. So it's not working very well. But, when we used before, so some years ago, the work with Telmo Mejia Garcia showed that nitric oxide can regulate cell survival using purified neuronal cultures. So you can see here, and when you, in this case, it's a model in which when you remove the medium, change the medium to fresh medium, you have an intense cell death, as you can see here. And this is completely blocked, this cell death, by SNAP, which is a nitric oxide donor, we say now a no donor, and as you can see here, and also that this effect of SNAP could be blocked, completely blocked by inhibitors of the PI3 kinase pathway, as you can see here. So this PI3 kinase leads to AKT phosphorylation. This is an extremely important kinase. As you can see here in this diagram, AKT can be phosphorylated by PDK1 and mTOR2, and all these three enzymes are anchored to the membrane in PIP3, so and can phosphorylate it in two sites of AKT. The two with serine 473 and threonine 308, and this activate different, as we call, multifunctional nodes. That's FOXO, GSK3, and TSC2, and it can activate the mTOR pathway here, and this is responsible for several of these nodes are responsible for different cellular functions as survival, proliferation, metabolism, growth, and others through different kinase systems. And the other actor here in the study is the nitric oxide system. So you have here the actors or the system, as you can see. L-arginine is the precursor of nitric oxide. And you have here the nitric oxide synthase producing NO and citrulline in stoichiometric manner. And here you have the, the pathway, the canonical pathway for activation by nitric oxide, as pointed by Dr. Hannes. As you can see here, nitric oxide can activate soluble guanylyl cyclase and produce cyclic GMP, which activates protein kinase G. It's a cyclic GMP protein kinase, and this can phosphorylate different substrates, including transcription factors that can lead to changes in gene expression. Another actor in this nitric oxide scene, as you can see here, is the arginine, L-arginine transporters. You have many subtypes, and the most important is the CAT1, is a cationic arginine transporter type 1. So some years ago, Telmo also showed that nitric oxide regulates AKT phosphorylation and nuclear translocation in cultured retinal cells. As you can see here, I want to show some of this data. The SNAP, which is the node donor, can promote the phosphorylation of AKT in both sides, 473 and 308. And this is also mimicked by, this is a very interesting point, by L-arginine, the substrate of nitric oxide synthase. By L-arginine can promote this phosphorylation of AKT, and this is blocked by PI3 kinase inhibitors. And you can see here, you have this at the right side of the slides here, you can follow the transduction pathway taking form. So the next, we will see here that L-arginine can cause phosphorylation of AKT in a dose-dependent manner, okay, the same amount as SNAP. And the effect of L-arginine is completely blocked by L-name, which is a no synthase inhibitor. And so this effect can be produced by SNAP or L-arginine producing no activation of PI3 kinase and phosphorylation of AKT. So the next, you can see here that this effect depends on the canonical pathway. As you can see here, soluble guanylyl cyclase and PKG are involved. YC1, which is a guanylyl cyclase stimulator, can promote the AKT phosphorylation. This is completely blocked by PI3 kinase inhibitors. And also SNAP, which is the node donor. The effect of SNAP can be blocked by ODK, LY, and KT. So this is showing that the soluble guanylyl cyclase and PKG are involved in this pathway. So now with unpublished results, also by Telmo, show that the SNAP or L-arginine can promote this phosphorylation of AKT in a calcium-dependent manner. As you can see here, this effect is blocked by BAPTA or HGTA, which are calcium chelators. And so we started to check how is calcium working in this pathway. So the first thing is that the effect of AKT phosphorylation depends on Cal modeling. The effect of SNAP or L-arginine is completely blocked by 3 different Cal modeling inhibitors, as you can see here. So the effect of SNAP is calcium dependent and involves Cal modeling to phosphorylate AKT. And then the effect of AKT phosphorylation is also CAM kinase dependent, which is the effect of SNAP is completely blocked by KN93 which is a kinase inhibitor, as you can see here. And so the effect involves calmodulin and kinase, maybe kinase 2. And as you can see here, SNAP or L-arginine can also phosphorylate kinase 2 here, as you can see. And this effect is obviously blocked by the inhibitors, the kinase inhibitors. And also you can see here in the fluorescence, immunofluorescence, that SNAP can increase phosphorylation of kinase 2, as you can see here. And also this effect of AKT phosphorylation is SRC dependent, so it's completely blocked by PP1, which is a SRC inhibitor. So the effect of SNAP, the effect of YC1 or L-arginine are all blocked by PP1. So this is involving also SRC. So we went on to see how calcium is or actually nitric oxide regulating calcium. So to students, PhD or a postdoc student Ivan Domic and the PhD student Aline Duarte Silva went to study this effect of nitric oxide in using transfecting the cultures. He mixed cultures from the retina, the embryo, with this compound that's very well known now, GCAMP. It's a calcium sensor. And you can see here that in this same, actually we cannot see, it's not working. Let me see. Oh, a moment. I'm returning. It's very slow. It's not working the videos. No. Trying to return. One more. So you cannot see here, but when you stimulate, you can transfect neurons in this culture. When you stimulate with glutamate, you have a very nice response. This get red and appears all the neurites here. And also, it's a pity, it's not working. You can see this was amazing data. So you can see here intracellular calcium observed after increasing L-arginine concentration. So it's not showing here. You have a very big and increase in calcium in these cells. This is a glial cell transfected with the GCAMP. And this is concentration dependent, as you can see here, with different concentrations of L-arginine. And this effect, this increase, is dependent on nitric oxide synthase, NO. As you can see here, this is blocked by 7-NI, which is nitroindazole, which is a NOS inhibitor, as you can see here, and also by no, it's not working well. Returning, no. One more, please. One more. And one more. So it's not showing here. And this effect is also blocked by PTIO, which is a no scavenger. And we can go ahead next. The calcium increase depends on, this is very interesting, on L-type voltage-dependent calcium channels. As you can see here, it's not working. No. No, returning. Well, and the effect is also, well, it's very important to see that the nifedipine, which is a calcium channel inhibitor specific for L-type voltage-dependent calcium channels, and blocks completely the effect of L-arginine. So the effect is involving this type of calcium channel. The effect is also dependent on guanylyl cyclase and PKG, so the canonical pathway for cyclic GMP. As you can see here, ODK, which is a guanylyl cyclase inhibitor, blocks the effect. And also KT, which is a PKG inhibitor, also blocks the effect. And YC1, which is a guanylyl cyclase activator, promotes a PKG-dependent effect. As you can see here, is more long-lasting than arginine, and is completely blocked by KT, which is a PKG inhibitor. And you could, if possible, see the videos here. It's not working, sorry. So then, concluding here, we have a pathway that's leading to AKT activation by L-arginine. So L-arginine enters the cell through cationic amino acid transporters and activate NOS, which is arginine is a substrate of NOS. So it increases the production of NO and citrulline. And this can be blocked by 7-nitroindazole. Also, SNAP, which is a no donor, also can produce NO and activate the canonical pathway, which is soluble guanylyl cyclase and PKG. PKG can be directly activated by YC1. And this, in some way, we don't know, is increasing the activity of calcium channels. And the calcium channels are the L-type dependent calcium channels. So this is inhibited by nifedipine. And so in this way, you have an increase in calcium, a very huge increase in calcium inside the cells, in glial cells of the cultures, and also in neurons, but mainly in glial cells. And this calcium can activate calmodulin, and this is blocked by calmodulin inhibitors, and calmodulin can activate, in calcium calmodulin complex can activate CAM kinase, and CAM kinase are inhibited by these compounds here. And all these inhibitors block the phosphorylation of AKT. So this CAM kinase in some way activates SARC. This is inhibited by PP1, and PI3 kinase and AKT. So this is the pathway linking L-arginine to AKT, but we don't know very well several steps, but it can be studied in the future. For example, the most important for us is how PKG is controlling or regulating calcium channels. So this is the last slide. And then next, I want to thank the people in the lab, now Laboratory of Cellular Neurobiology from Nacional University, Ivan Domicis, postdoc, Aline Duarte Silva, PhD student, and the other students here, technical support by Sarah Rodriguez, and collaborators now in the University of Tübingen, very important collaboration now with Robert Feil in the lab, and Aline, for example, is going to sandwich PhD in his lab. And also we have a collaboration for years with the University of Porto, João Helvas, and Camila and Renato. Renato will talk in a bit. Working there at the University of Porto. They were former PhD and postdoc students in my lab, and also the collaboration of Susan Amara that gave some of the including the GCAM probe we used. She works in the National Institute of Health. So this is Niterói. I hope you can visit there. It's a very nice place. And we have a new building there. So you are invited to go there when it's possible. Thank you very much.
Thank you, Roberto. Right on time. So I will give the words to you so you can present Renato and finish the roundtable, okay? Thank you.
So it's Renato there. Hi, Renato. Here we are again. So
let's see if it works.
Oh, okay, great. So Renato, as I told before, he's got his PhD in neuroscience in 2012 in my lab, and he was very interested in tyrosine kinase controlling microglial function in the CNS. And now his recent publications show critical roles for tyrosine kinase and RhoGTPase signaling in controlling microglia-dependent inflammation neurodegeneration. And he's now present a postdoc fellow at the Institute for Molecular and Cell Biology of the University of Porto. Thank you. You can go ahead. Thank you.
I would like first to thank you, the organizing committee, especially Roberto, for inviting me to present my work and the work that we've been doing in the lab for the last couple of years. And for me, it is particularly interesting because, as Roberto said, we worked together for over 10 years, and participating with him in this meeting feels like the son is uniting or meeting the father again after a long time. So thank you, Roberto, for this. And the talk today will be most related to microglia and how this little population of cells in the central nervous system can interact and modulate neuronal function with a particularly, and we are particularly interested in how RhoGTPases in the microglia can modulate this, let's say, crosstalk between the glia and the neuronal compartment. So next, please. The microglia, next. They are the resident immune cells of the central nervous system. So they are derived from the yolk sac early during development, and they share a different embryonic origin from other CNS cells, but nonetheless they can engraft the central nervous system or the developing central nervous system very early, like they contact neurons and the initial synapse formation much earlier than other CNS cell types like astrocytes or oligodendrocytes. So they must have a very preeminent role in regulating neuronal function since early the beginning of the development. And this has been shown, for instance, in the case of refinement of neuronal circuitry during development. So these cells are very important in these aspects. And this, as is written there, they are a tiny population of CNS cells. They constitute, depending on the region of the central nervous system, of course, but they usually represent 5 to 10% of all brain cells, and they're critically involved in the preservation of CNS homeostasis. And I think we can, let's see if we can convince you of some of these aspects during the talk. Next, please. So the classical neuron microglia crosstalks involve, of course, the so-called microglia surveillance function, microglial surveillance function, and the crosstalk between these two cells may occur between two major aspects. It's like the secretome of microglia, so the things that microglia can secrete to the environment and modulate neuronal function, or can be via contact-mediated functions as represented here. And this also has important functional outcomes because microglia as a main immune cell can mediate phagocytosis effects and also promote a thing called synaptic pruning, in which these cells can engulf and modulate the number of synapses in the central nervous system. And this process occurs both during development and in the adult central nervous system. Next, please. So these are the called microglia housekeeping functions and how they can interact with neurons via contact mediated or secreted things, but as a classical immune cell or myeloid cell, they also undergo the so-called immune activation when they are presented with some stress in the system, like for instance, during injury or during infection with a pathogen, or even during neurodegenerative disease. And what happens usually is that these cells undergo dramatic morphological and transcriptomic changes, and they transit to a classically named inflammatory response, in which they can release massive amounts of cytokines, mainly pro-inflammatory cytokines and other chemokines, and also produce and release reactive oxygen species and also glutamate. In this pathway of, for instance, pro-inflammatory cytokines and glutamate will be particularly relevant for the talk today. And the other aspects that they can also, these cells can also modulate is like the debris clearance in the central nervous system, also during neurodegenerative diseases like multiple sclerosis, or in the case of engulfing and clearing myelin debris, or during, for instance, amyloidosis or an Alzheimer's disease, in which these cells can interact and also clean up amyloid debris. Next, please. So, a unifying role in these housekeeping and activated profiles of microglia is obviously the cytoskeletal dynamics. All these processes that microglia perform likely relates with a very dramatic and dynamic reorganization of the cytoskeleton. And the premise here in this talk will be that disrupting the microglial cytoskeleton, or the dynamics of the cytoskeleton, should have a huge impact in brain homeostasis and, of course, in the crosstalk between microglia and neurons. Next, please. So, the way in which we interfere with this microglial cytoskeleton dynamics will be modulating these proteins called RhoGTPases, which are classically known as the master regulators of the cytoskeleton, mainly the actin cytoskeleton. And the Rho family is composed by typical members, the classical typical members, and atypical members. And for the talk today, I'll be mostly involved in the regulation of the Rho family, particularly the Rho-A, which is a prototype member of the family. But there are other members, the classical members or typical members, like RAC1 and CDC42, and the others are typical members. Next. So the typical members of this RhoGTPase signaling, actually they work, this typical RhoGTPase work as binary switches cycling between active or GTP form bind and inactive, which are bound to GDP. And this cycling is obviously modulated by GAFs and GAPs, and further regulated by RhoGDIs that can sequester the RhoGTP form away from the activation site. And of course, when these RhoGTPases are bound to GTP, they are in the active form, they can perform several cellular functions by activating a number of proteins called effectors. Next, please. So this is a classical view retrieved from the paper from Alan Hall in Science in late 90s, in which they his group studied the classical function of these RhoGTPases in modulating the cytoskeleton. And at that time in fibroblasts, what was established was that the Rho signaling is classically associated with the formation of these structures called stress fibers, then that can culminate with the formation or the recycling of focal adhesions in cells, whereas RAC1 function mostly relates with the formation of these structures called lamellipodium, and the function of CDC42, for instance, is mostly related with the formation of philopodia, which are these spike-like structures in the cells. Next, please. Although these were the classical functions established for this RhoGTPase, it's now known that actually these RhoGTPases, particularly RhoA, work as integrators of signaling from the extracellular space to the intracellular milieu. And actually, RhoA and other members of the typical members integrates a myriad of stimulation from the ECM, from GPCRs, tyrosine kinase receptors, EGG or IG superfamily immunoreceptors, adhesion signaling by caderins, and also mechanical stimulation integrating to the intracellular pathways. And of course, this all integrates with the regulation of cytoskeleton organization, but also they are also involved with gene expression, membrane trafficking, cell adhesions, cell cycle progression. It's a very important role of these RhoGTPases. Cell migration, very important, for instance, during neuronal pathfinding, cell polarity in epithelial cells, and also in the case for microglia and other immune cells, the secretion and endocytosis of cell-bound membrane receptors, or the secretion of cytokines or pro-inflammatory mediators. Next, please. So the focus will be mainly in RhoA, and as we can see in this slide from the Brain RNA-seq published by the Bembar's group, we can appreciate that RhoA is ubiquitously expressed in the central nervous system. And although they are the mRNA species for RhoA, both in humans and in the mice, are highly expressed in microglia or in myeloid cells in the central nervous system, they are also expressed in all other cell types in the central nervous system. So, next, please. The only way to certainly study the role of these proteins in the central nervous system, in any other cell type in the central nervous system, is, of course, to use approach that integrates cell type specific modulation of these cells. And next, please. And the way that we do this in the lab is basically using Krylok's technology, in which we have to cross actually a RhoA floxed mice with another mice which carries the Kre-insertion cassette, the Kre-R cassette within the CX3CR1 promoter, which confers microglia specificity in this case. And we cross these two mice line and produces a third mouse line, in which we call the RhoA conditional knockout mice. That, next please, when we give these mice tamoxifen, we will ablate RhoA in microglia in a temporal dependent manner. It's very important to note here that this system is completely cell specific, so we only work with the deletion of RhoA in microglia, in this case in adult microglia, because we give tamoxifen to the mice in the early adult stage, and all other cell types in the brain will have the RhoA expression preserved. So the protocol is basically we give tamoxifen to these mice to ablate the gene between P28 and P30, and analyze the brains and do other behavioral tests 2 to 5 months later. And the results will be that I will present today will be mostly within this time window. Next, please. So the first thing is that to demonstrate that actually the knockout is working. Next, please. And another. And next, please, again. Next. Okay. So we used different approaches to show that the RhoA was ablated in microglia using qPCR in flow cytometry sorted microglia, and also demonstrated the loss of RhoA protein in these microglia, but not in the close relatives, the macrophages. So it seems that, okay, that the system is working, and upon tamoxifen administration to these mice, we can ablate and disrupt and stop expressing RhoA only in microglia in this system. Next. First, the first phenotype that we saw was we looked for microglia and found that when we remove RhoA, we have a decrease in different brain regions of the microglial numbers in these mice. And this decreased number of microglia was associated with an increase of necrotic cell death in microglia, but not apoptotic cell death. Next. Please, next. One more. Yeah, okay. And this loss of microglial viability was associated with a spontaneous activation of microglia, because what we found is that when we remove RhoA from these microglia, they start producing, also secreting larger amounts of TNF and IL-1 beta, which is a classical pro-inflammatory phenotype, but also they start to produce large amounts of reactive oxygen species and integrate these oxygen species with the activation of NF-kappa B, and this inflammatory module culminates with the increased release of glutamate from this microglia when RhoA was not present. And this phenotype seems more or less specific for RhoA and is not compensated by the other closest family members, RhoB or RhoC, because when we knock down RhoB or RhoC, we cannot mimic the phenotype. So the phenotype seems quite specific, and it's not compensated by other RhoGTPase, at least the closest family members. Next, please. So this, let's say, spontaneous activation of microglia when in the absence of RhoA, in this pro-inflammatory activation and polarization, obviously probably has implications for the neurons. And the next thing that we did was to actually see if the ablation of RhoA was impacting the viability of neurons and synapses. And what we did first was to knock down RhoA in microglial cultures and then collect the media from this microglia without RhoA and expose them to neurons. And what we observed was, comparing to control cells in which RhoA was not knocked down, the knockdown of RhoA in microglia induces a synaptotoxic effect in these hippocampal neurons, and this synaptotoxic effect could be prevented if we incubate this conditioned media from microglia with a glutamate receptor inhibitor, namely ionotropic glutamate receptors inhibitor. And in vivo, in the mice, what we observed was that when we looked at the synapse, the synaptic plasticity in the hippocampal CA1 to the Schaffer collateral synapses, and studied the LTP in this circuitry, we observed that the ablation of RhoA in microglia was sufficient to largely impair the LTP in these mice. Next, please. So synaptic plasticity was impaired, and this synaptic plasticity impairment was associated in these mice with a decrease in the number of excitatory synapses in the hippocampus and also in other brain regions, and this loss of synaptic plasticity and loss of excitatory synapses was obviously had an impact on behavior, because these mice presented a clear deficit in recognition memory, which is a common feature in many neurodegenerative disease. Next, please. So, next. So the conclusion in this first phase is that first, in the microglial compartment, RhoA sustained microglial survival and restrains microglial immune activity. And the last thing is that the loss of RhoA in microglia leads to a neuronal dysfunction. And I will skip the second part, please. Go. Next. How this happens? Next. Basically happens in a way that RhoA regulates intracellular signaling controlled by CSK and the tyrosine kinase SRC. And I will skip this part because of the time. Please, next. Next. And we could show that when we remove RhoA, we modulate the signaling from CSK to SARC, we decrease SARC and activate SARC, and this mimics the phenotype in the mice. Next, please. And we also could prevent the phenotype if we block SARC in the mice that don't have RhoA in microglia. Next, please. Next. There, if we use a clinically relevant SARC inhibitor in the microglial RhoA knockout mice, we obviously decrease the activation of SARC that is shown here. And more importantly, we can prevent the loss of synapses and also prevent the behavior, the memory deficits that was found in these mice. So the pathway from RhoA to SARC activation is operative in the in vivo to lead to synaptic dysfunction and memory deficits. Next, please. And this also occurs if we block SARC in vitro. We also can prevent the pro-inflammatory polarization of microglia triggered by the reduction of RhoA. Next. So the conclusion for the second part, very briefly, is that blocking SARC largely compensates the deficiency of RhoA in microglia. And the last part is related mostly with neurological phenotype. Next. Showing next, showing that these features of synaptic loss, memory deficits, microglia hyperactivation, and memory deficits are common features in many CNS neurological diseases and disease states, and also in Alzheimer's disease. So we asked if this sort of RhoA signaling was more or less altered in Alzheimer's disease mice. Next. And very briefly, we showed that using the classical model of amyloidosis, APP-PS1 mice, that this module of signaling, RhoA, CSK, SARC, was altered in these mice, that in microglia from these mice, the activity of RhoA was decreased, the expression of CSK was also decreased, which accompanies the decrease in RhoA activity, and this leads to a hyperactivation of SARC. So this pathway is also operative in a mouse model, let's say mouse model of Alzheimer's disease. Next. This can be reproduced in vitro by amyloid oligomers, that if we expose human microglia to these amyloid oligomers, we activate this pathway by the reduction of RhoA. Next, please. And surprisingly is that the amyloid was actually modulating RhoA, but in the mice that don't have RhoA in microglia, they develop a spontaneous amyloid phenotype.
Excuse me, you have more minutes, okay?
Okay. They have more toxic amyloid species like A beta 140 and A beta 142, and also produce a small amyloid plaque-like phenotype. So the single fact that removing RhoA from adult microglia, the mice develop a neurological phenotype associated with amyloidosis. Next, please. So in this signaling pathway from RhoA to SARC was controlling the amyloid and A beta production in neurons, and we can see this by showing that if we take out the conditioned media from microglia without RhoA and expose them to neurons again, it's synaptotoxic, but also increases the production of A beta autochronally by neurons. And if we block SARC or if we block TNF in the background of RhoA, we can prevent this increase in amyloid production by neurons. Next, please. Next. And this also occurs in vivo if we block the activity of SARC using, again, that clinically relevant inhibitor called the AZD in the background of the RhoA knockouts. We can prevent the increase, we can decrease actually the production of amyloid, the production of toxic amyloid species, mainly the 130 and 142 amyloid peptides, and also we can clearly prevent the accumulation of oligomers to produce amyloid plaques. Next, please. So what's really happening, what we think that we figured out is that in the steady state condition, we have ideal activation of RhoA to sustain the expression of CSK, and this leads to steady state or normal activity of SARC. When SARC is not hyperactivated, we have this, the microglia circuitry of immune activity normal, and we can proceed with normal brain function, and we don't have amyloid plaques in the brain parenchyma. We have normal LTP and normal number of synapses, and this culminates with intact neuronal performance and memory performance. When we remove RhoA only in microglia in the system, we lose the capacity of restraining microglia immune activity because we decrease CSK expression, which leads to hyperactivation of SARC. And this hyperactivation of SARC engages in a pro-inflammatory circuitry involving NF-kappa-B activation, production of and release of TNF, autocrine and paracrine loop between TNF and glutamate. And when glutamate is released in high amounts next to the synapse, it's highly synaptotoxic and also induces the accumulation and production of A-beta by neurons. This accumulates with amyloidosis, LTP impairment, loss of synapses, and a neurological phenotype characterized by memory loss. And actually, we think it's this RhoA mice is more likely related with an animal model for amyloidosis, or more or less an Alzheimer's disease-like animal model. And to round up, I would like to thank the lab. First, the generosity of João Helvas and the long-time collaboration with Camila, Portugal, and the students that also helped me take out the work, Arturo Rodrigues, Thiago Almeida, and Joana Moreira, and all other members of the GCB, the international collaborators, of course, Roberto, as he mentioned before, and other collaboration FDI3S with the addiction biology group, Teresa Sumaviel and Teresa Canedo. And thank you.
So thank you very much for this great presentation. And now Ricardo is there. I think we have time for questions. Not many, but a few. So you can go ahead. And I think we have also some questions here in the chat.
Right, Roberto. There is one question here to Dr. Schmidt about the bifurcation. If the bifurcation process persists in adulthood in neuronal plasticity, such as condition of restoration of function after lesion in CNS. Could you listen? And I would like to add to that, you show data on cranial neurons and peripheral neurons. If this bifurcation with the deficit in the protein that you showed has problems with neurotrophin signaling, like CNTF in the cranial neurons and NGF in the peripheral neurons.
Yeah, thank you for these questions. First, to the question of Valeria Sosa. So the bifurcation process is laid down during embryonic development. So in developmental time window between embryonic day 10 and E13. But when we observe the bifurcational phenotype with the Cy1 YFP reporters, then we see that the bifurcation is stable also to later stages and to adulthood. So it's not reformed if you have the loss of bifurcation due to the absence of the CNP-induced cGMP signaling. It's not later that the second branch grows out, so it stays with these turns only in one direction. An interesting question would be what happens if you have a lesion later on. Maybe you damage the dorsal root. I think the problem is that also these DRG neurons lose their ability to regenerate into the spinal cord, but another problem is that also the expression of the ligand and the receptor is very dynamic. So this embryonic pattern, which we see where we have CNP in the dorsal spinal cord, disappears very quickly. So we have then more a punctate pattern where only some spots over the whole spinal cord still express the ligand CNP. And we showed by X-Gal staining and also by Western blot against the receptor NPR2 that then at later stages at P75, only 10% of all DRG neurons still express the NPR2 receptor. And therefore, I think just the proteins wouldn't be there in the DRGs at least to induce a new bifurcation during regeneration. And for other parts in the brain, we don't know whether they actually use this signaling cascade for their branching. We chose the DRGs and also craniosensory ganglia because they have a very stereotyped pattern of branching where all the neurons have the same pattern, and it's easier to follow this in comparison to the complex branching pattern which you could observe in central neurons. And the second question is about the NGF-TrkA signaling. So what we found out in cooperation with the lab of Gary Lewin, who measured the ventral root potential after dorsal stimulation, is that in the knockout mice which lack bifurcation, you see a reduced ventral root potential. And this also goes along with a decreased branching behavior in the TrkA positive nociceptive neurons. So there is also a decrease there.
Many thanks.
Thank you.
Before asking a question to Roberto, I would like to ask a question to Arturo. Sorry?
I have one for Arturo, but you can go ahead.
No, go ahead, please.
Arturo, okay, thank you very much. So you showed that D-aspartate can activate mTOR. So it's a very simple question. And how is it done? Is it involving AKT pathway?
Yes, well, it's a calcium-dependent. The whole signaling starts by the reverse function of the sodium-calcium exchanger. And then it's also, if you go and block with guanine, you kill the response. So that's BI3 dependent. Yeah.
Thank you.
Arturo, I would like to ask a question to you also. We have shown that glutathione, that's the major antioxidant in the glia cells, they have a selective calcium influx in glia, but not neurons. Have you seen anything related to glutathione and the glutamate transporters to see this regulation that you show about calcium and glutamine synthase?
What we have done in that regard is only start to characterize the cysteine glutamate exchanger, the XCT. Turns out that it forms like a complex with GLAST and with the SNATs. We're actually working on that, and we have some sort of related thing when we go with arsenic. And we check for production of glutathione, and it was not a very strong response. So we don't know, but we're still working on that.
Excellent. Okay, Roberto.
Yes.
I would like to ask a question to Renato also, if possible. Renato, excellent data. I mean, I really enjoyed the images of Dr. Hannes Schmidt, also amazing pictures that you showed. Renato, there is this kind of multiple microglia phenotypes, M1, M2, and others also. Do you think that this kind of phenotype that you described with the Alzheimer's linked and excitotoxicity, could you revert this physiologically by the other types of CDCs and RhoA proteins also? You have several proteins that are involved with cytoskeleton, correct? So maybe could you go to healthy and pathological states, just modifying the prototypes in the biochemical phosphorylation of the members? That's the question, if you could comment.
Yeah, okay. what I can disclaim about this is actually that the phenotypes for the typical RhoGTPase, we are also have fluxed the RAC1 in microglia, and hopefully can send a manuscript to publication in the next few months, and also flux the CDC42 in microglia. And what I can say is that the phenotypes are completely unique for each one. There is no sharing of the phenotype. So when we remove RAC1, for instance, we don't have a neurological phenotype and neurotoxic phenotype like we have in the RAC1, but we have behavioral deficits as well. And this majorly relates with growth factor signaling that RAC1 is regulating in microglia that goes to the neurons, mainly GDNF, for instance, glial-derived neurotrophic factor for the RAC1, and the CDC42 have a much ampler phenotype because we impact several things in the synapse when we remove CDC42 in the microglia. But again, they are not a synaptotoxic phenotype, and it's not a phenotype that is related to RAC1. So both the three phenotypes are very distinctive, and they, surprisingly for me, they share very little similarities between those three, which again shows that the RhoGTPase signaling is very cell type specific, because if you look for the macrophage system or in peripheral immune cells, the phenotypes are very well described, and RhoA and RAC1 share many similarities there, but in the microglia they are very distinctive.
Wow, excellent. And Roberto, the last one for you. Question from...
I have a question. Are you hearing me? I have a question to Renato and another one to Hannes, if I can. Can I?
Sure, go ahead.
Okay. Renato, just to, I don't know if I got everything from your talk. It's very interesting. We are very interested in SARC, still very interested in SARC. And you said that the glutamate is released. And do you think that what you have, is it dependent on SARC, and how?
Okay. I have a paper published around that, and it relates with SARC and calcium as well. So we don't know how exactly how calcium regulates SARC, but calcium is upstream of SARC in this context. And what SARC does is, what we believe is two things, depending on the context. Like if you have inflammatory context, goes from one pathway, and if you have more like a hypoxic or neurodegenerative context, may go for around. But major two pathways that SARC activates inflammation via NF-kappa B, and this inflammation produces and releases TNF, and TNF autochtonally stimulates the release of glutamate via connexins. And the other phenotype is that somehow SARC is regulating RAC1, and this can be via activation of RAC1 GAFs, which is described in other systems. And RAC1 is a main component of the NOx complex, which produces reactive oxygen species. And we don't know exactly the molecular signature, but hydrogen peroxide can oxidize connexins and other probably CX- transporters, and also promote the release of glutamate from microglia. Differently from astrocytes, this microglia, at least no one described that so far. They don't have a mechanism for vesicular release of glutamate, but they have major calcium-dependent glutamate release machinery. It's not a SNAD-dependent complex so far, but of course, calcium is modulating everything in terms of glutamate release. Let's say yes. Calcium in the upstream of the cells.
Yes, it's interesting because in our system also SARC is downstream of calcium signals. So maybe from L-arginine and nitric nitric oxide. So is it nitric oxide working in your system as well?
We never tested the nitric oxide because actually when we activate these cells, we have huge amounts of nitric oxide coming from ions. We don't have the other isoforms, at least physiologically relevant levels of the other isoforms, but the isoform 2 is triggered there. And we don't know, we never looked into a nitric oxide, but probably it's participating in the activation of SARC somehow, yeah.
Yeah. Thank you. I have another question. Ricardo, you have another question? I have one for Hannes. This is just a general one. Okay.
No, I'm all right. There is one question for you, Dr.
Yes.
Marcelo
But first,
Coscienza. Sorry.
okay.
Do you have another one?
No problem. Go ahead.
Marcelo is asking you if arginine could activate calcium channels directly, or if it's independently of no formation.
Very good question, Marcelo. And we think that the L-arginine is promoting this calcium increase through NO, and mostly by NO, nitric oxide, because it's blocked by NOS inhibitors, etc., 7 and PTIO, etc. So it's more than 80% inhibition. So but maybe you have a 20% that is not blocked, and you have this in our system, something that is not no dependent, but we don't know what is, if it is L-arginine directly, or you have many things that are formed through or from L-arginine, like other things like agmatine, etc. Okay, thank you. And I have a question to Hannes. It's just a general question. I don't know if I got from your wonderful talk, is that the CNP is well known, that is it distributed in other areas of the main areas of the brain, and is it also, you have papers and data showing that the cyclic EMP system through PKA and through APAC also participate in axon growth, etc. Is it interfering in your system? They can be antagonic or working in the same way. What do you think? You have two questions: CNP around the brain and cyclic EMP in the axonal growth.
So, starting with the first question about the brain localization of CNP, it's very interesting. So we have now this LAXI reporter mice where we can actually map the expression also in other areas of the brain. And we find that it's highly expressed, for instance, in the hippocampus, but also in the cortex, so in many areas actually. And this is quite funny because the one ligand for the NPR1 receptor, BMP, would receive the name because it's a brain type natriuretic peptide. But when we look at CNP, it's also very much expressed in the brain. We don't know yet from other phenotypes due to a loss of CNP in the brain, but we are currently looking into that with respect to the hippocampus, whether there's something. And the second question was an interaction with the cAMP system. And actually, this was a starting point for us because there were these papers by the lab of Muming Pu in the late 90s already, which showed that the response to certain guidance factors is affected by the a ratio between cAMP. So, and there are, of course, phosphodieesterases, which are bifunctional and could interfere at the same time with CNP and cGMP concentrations in these cells. In the first reports, it was suggested that an increase of cGMP could, for instance, convert a repulsive action by a repulsive guidance cue to an attractive action. And later on, this was modified that it's really the ratio between cAMP and cGMP generation which affects this response to guidance cues. So there are certainly a crosstalk between these two factors, but with respect to the sensory axon bifurcation, we have no data available that shows that cAMP is involved there. We found out that the phosphodiesterase, which is mainly responsible for the degradation of cGMP there, is PDE2A. But if we knock out PDE2A in the DLGs, we don't see a bifurcation effect. So this seems that the regulation of cGMP by PDE2A is not so much then as a consequence for the bifurcation phenotype.
Okay, thank you. Great. Okay.
Roberto, the other questions that were here, Renato answered, and I put them in the YouTube. So I think we are ahead, like 20 minutes more, and we have to finish here to the afternoon session that will be a closed one. Maybe you can finish this session, okay?
Yes, I just want to thank the speakers for the wonderful talks, and thank you very much. And I hope you all in the audience have fun and enjoyed this very nice roundtable. Thank you very much.
Thank you.
Thank you all.
Thank you.
Arturo, I will write to you, okay? We have lots of things to talk. Say,
okay,
bye.
Thank you.
Bye, Renato. Bye, Dr. Hans.
Bye-bye.